Artificial loss in precision, compared with an unadjusted analysis. https://www.fda.gov/media/148910/download 18. Review example 2, AI-generated digital twins and prognostic covariate adjustment, continued. Summary of FDA's feedback/questions to the sponsor. No objection if it is implemented in the Phase 2 trial as an exploratory analysis. Its use as the primary analysis in the Phase 3 trial may be acceptable. Provide details. For the Phase 3 trials including data analysis plan etc. Is the training dataset for the prognostic model representative of the population the sponsor plans to enroll? Since generative AI will be used for prognostic score generation, ensure the same patient retains the same value if the process is repeated multiple times. Suggestion of using explainable AI techniques to identify key contributing features. For the prognostic score. 19. Review example 3, ML-based patient population selection. Section 1.1 Fact sheet of Kineret registered. Anakin EUA review https://www.fda.gov/media/163546/download. Louis TALCPT DOI 10.1002/CPT.3191. Patients in the Phase 3 trial, Save More, were selected based on a biomarker cutoff, SUPAR greater than or equal to 6 ng/ml. Intending to enrich the trial with patients at risk for progression to severe respiratory failure. SUPAR test is not commercially available in the United States at this time. Regulatory Challenge 20. Liu et al. CPT. DOI 10.1002/CPT.3191. Using ML to address the challenge, is there an alternative way to identify patients that are aligned with patients enrolled in Save More trial, SUPAR high? 21. Selected score, 3 out of 8. SOFA * score greater than or equal to 3. Smoking history, active or past. Blood hemoglobin less than or equal to 10.5 g/dl. Medical history of ischemic stroke. Severe disease by WHO criteria. Age greater than or equal to 75 years. Blood urea greater than or equal to 50 mg/dl and/or medical history of chronic renal disease. Neutrophil to lymphocyte ratio, NLR, equals 7. *SOFA, sequential organ failure assessment. Anakin EUA fact sheet. U.S. fact sheet of Kineret registered drugs at FDA, FDA approved. Drugs 22. Challenges in the application of AI/ML. Bias. Generalizability. Opacity. Privacy. Security. Other. Disparity/fairness. Trustworthiness issues. Figure generated by Chi using AI, DALL-E 323. FDA AI discussion paper 24. AI/ML activities in Office of Clinical Pharmacology, OCP/FDA. Innovative Data Analytics. IDA, Program. For FDA Clinical Pharmacology Machine Learning. Fellowship, Checkout. https://www.zintellect.com 25. AI/ML research in OCP. Landscape analyses. Application of machine learning in drug development and regulation, current status and future potential. PMID, 319259555. Landscape analysis of the application of artificial intelligence and machine learning in regulatory submissions for drug development from 2016 to 2021. PMID, 3570790. Methodology exploration. Long short-term memory recurrent neural network for pharmacokinetic pharmacodynamic modeling. PMID, 332109994. A novel approach for personalized response model, deep learning with individual dropout feature ranking. PMID 331049924. Application of machine learning-based methods in exposure response analysis. PMID 352751315. Measurement and mitigation of bias in artificial intelligence, DOI.org/10.1002/CPT.3117. Methods for preventing prediction for out-of-scope data. Interpretable/explainable ML. Large language model for document editing. Application for therapeutic optimization/individuation. Ongoing research, use ML to predict prognosis or treatment outcome, both efficacy and toxicity. Multiomics data for precision medicine, PMID 3796585. Published. Ongoing www.fda.gov 26. Quantitative Medicine Center of Excellence. Clinical trial. Simulations. And in silico. Predictions. Pharmacostatistical. Models. Mechanistic. Modeling. Biomarker endpoint. Development. Machine learning and artificial intelligence. CDERQM CoScope 27. Take-home messages. AI/ML can play an increasingly impactful role in drug development. Improve the efficiency/probability of success of drug development. Advance precision medicine. Both opportunities and challenges lie ahead. More education, research, and collaboration are needed to move the field forward. Figure generated by Chi Lu using AI, DALL-E3FDA.gov/CDERSBIA28. Challenge question number 1. How can artificial intelligence enhance drug development? A. By improving the probability of success of drug development. B. By improving the efficiency of drug. C. By advancing precision medicine to tailor treatments to individuals. D. All the above. FDA.gov/CDERSBIA29. Challenge question number 2. True or false? Machine learning is considered a subset of AI. 30. Acknowledgement, Ruhao Huang, Hao Zhu, Menglun Wang, Lixia Zhang, Yanning Wang, Chao Lu, Yudao Gong, Daphne Jean, Yuanfang Guan, Huang Chen, Sebastian Thrun, Kair M. Elzarad, Tala Fakhouri, Matthew Diamond, Shumei Huang, Isam Zina, are current and past fellows.